Genetics problem number 15 says in Cocker Spaniels, black coat color is dominant over red and solid coat color is dominant over spotted. Give the genotypic and phenotypic ratios of the offspring for a mating between dogs heterozygous for both traits. The first thing to realize is that this is not a monohybrid cross. All the other ones we were looking at were one trait. Now we have two. Instead of 4 squares, if we have 2 traits, we need 16 squares. If we were looking at 3 traits, we would need 64 squares. And you can see that very, very quickly it becomes very, very complicated. So in Cocker Spaniels, we have black coat and red coat. Big B, little b. Dominant is black, recessive is red. We're still using the same rules as far as dominant and recessive uppercase, lowercase that we did before. But now we have 2 traits. So black coat is dominant over red, and solid coat is dominant over spotted. So solid, S is one you have to be very careful about making your S's different if you're going to use those. Spotted. Otherwise it's too hard to tell if it's a capital or lowercase. So solid coat is dominant over spotted. Give the genotypic and phenotypic ratios of the offspring for mating between dogs heterozygous for both traits. Okay, so we have 2 dogs that are heterozygous. Big B, little b, big S, little s, mated by big B, little b, big S, little s. Now the most difficult thing to do for these is to figure out what goes above these squares when you have 16. Luckily, you're not going to have to do any of these that are not heterozygous. I could make these homozygous dominant, I could make them homozygous recessive, and we could come up with all sorts of crazy 16 squares. But for this class, at this level, if you go on in genetics, you'll have to do all sorts of crazy squares. But for this class, we're going to keep it just straight heterozygous for both traits. That means that when we go through the process of meiosis and we go into gametes, we're going to have big B and little b, but big B has to match with big S, or it could match with little s. We don't know what's going to happen in meiosis. There's 2 different chromosomes we're dealing with. We have to account for all possibilities. Little b could go with big S, little b could go with little s. We don't know what's going to happen in meiosis. They're going to separate differently. So we have not just big B, little b to worry about, but we have big B combined with big S. We have big B combined with little s. Not just little b to worry about, little b combined with big S, little b combined with little s. Same thing for this parent. So we have 4 different possibilities for eggs and sperm with these 2 chromosomes. Imagine how complicated it gets with 23 chromosomes, like humans have 23 pairs. So for each of the boxes, we put one of these combinations because we don't know which egg and sperm it's going to be. So big B, big S, big B, little s. Little b, big S, little b, little s. Same thing here. Big B, big S, big B, little s, because we don't know which egg and which sperm are going to pair up. Okay, now once we get this is by far the hardest part of the whole process, because the down and across is exactly the same. We go down, we go across. Big B, big B, big S, big S. Down and across. Big B, big B, big S, little s. Here's the big S, here's the little s. Down and across. Big B, little b, 2 big S's. Down and across. Big B, little b, big B, big S. See, it's not so easy to do it. I'm trying to talk and write at the same time. Okay, big S, little s. Now let me fill in the rest of these without talking, because otherwise I'll get them messed up. You can predict as I go along what they should be, and hopefully I won't make any mistakes because I'm going too fast. All right, So when I look at these, we know that the traits are separate. We know that black is dominant over red, solid is dominant over spotted. So what we want to look at are genotypic and phenotypic ratios for all these. What I'm going to write out are not the genotypic ratios because we would have a bunch of those. This is one genotype. This is a separate genotype, but they have the same phenotype. They have a dominant black coat and a dominant solid coat. This is solid just like this is solid because each trait is individualized. So this is going to be black solid. This is going to be black solid. Let's see how many black solids we have. Black, solid, black, solid, black, solid. This is black but not solid. Black, solid, black but not solid. Black, solid, black, solid, not black but solid, not black but solid. Black, solid, nope, nope, nope. What we have here for black and solid, if we count the ones that I made the lines under, I have 1, 2, 3, 4, 5, 6, 7, 8, 9. I have 9 out of 16 black solid. Okay, now I'm going to come up with another signal. Let's do black spotted, because if there's no big S, it's going to be spotted. So I'll make a dot up here. Black spotted, black spotted. No, here's another black spotted. Okay, those are the only 3. 3 out of 16 black spotted. Okay, now we have no more black because we only have these 4 left, and they're all recessive for color, so they're all red. But some of them are spotted and some of them are solid. So if there's a big S, it's solid. So let's put a line through. We have, this is red solid. So red solid, red solid, red solid. 3 out of 16 red solid. And then there's only 1 left that is recessive for both. So 1 out of 16 red spotted. This is why when you pair 2 heterozygotes for 2 traits, that was my email, 2 heterozygotes for each trait, you always get a ratio 9, 3, 3, 1. And that's shown in your book, but they don't really go through why you get that. And this is why you get that. And it's going to be the same layout for any pair where they're heterozygotes for both traits. Now, if I did one where one of these was homozygous, either recessive or dominant, that's going to change this whole thing. And you're not going to get a 9331 then. 9331 is only if the parents are heterozygous for both traits. That's when you get that. And what the 9331 ratio means is that you get 9 that are dominant for both. Black and solid are your dominant traits. That's what's going to be most common. Black, solid, dominant for both. Each of the 3s is dominant for one trait, but recessive for the other. So in this case, a probability 3 out of 16, dominant for color, but recessive for solid. It's spotted instead. In this case, this is dominant, but this is recessive. So your 3s have 1 dominant, 1 recessive of the trait. You only get a 1 out of 16 probability of having both recessive. So for dihybrid crosses, the picture is a lot more complicated, and I'm only showing you if there's 2 heterozygotes. Otherwise, it would be a different layout. It would still be 16 squares, and it would still have the same one of each trait, but it could get a lot more complicated, and we would have to do a lot more practice ones of that if I was going to have you do 16 squares. With the time limitations we have, we just do this one type of 16 square, which is the 2 heterozygotes. You will have lots of different combinations of monohybrid, but your dihybrid, if you understand this 2 heterozygote situation with the 9331 results, you'll be where you need to be for what we're doing right now.